Hello, you're watching Bones and Stones, and what I have here for you today is a baby shark. I don't know what species it is, because when I asked the fishmonger, he said, I don't know, it's just a baby shark. There is a problem with this particular specimen, which is that it's been gutted, it's been cleaned so that the shark can be consumed, and that will limit dissection somewhat for obvious reasons, but there still should be organs, so let's take it to the table and see what we can find. So we have our shark ready to dissect as much as we can on this one, and I want to start by saying that the fishmonger said he rarely gets these in, and the reason for that is because you're not supposed to catch these around Europe or in the UK, so these are always imported from places where sustainable fishing is possible due to the populations there. I also don't in any way condone the eating of sharks for obvious reasons. I think about 50% of species are endangered, and if me buying this shark and dissecting it to show you how beautiful they are on the inside as well as the outside stops another person developing a taste for them, then I've done my job today. So let's have a look. We've got a lovely fins. We've got a caudal fin, a dorsal fin, and our pectoral fins that this shark uses for gliding through the water. And if you can see these little pits on the snout here, you can just about see these tiny little pits. These are the ampullae of Lorenzini, and these are what the shark uses to detect electrical currents and thereby sense other fish around it from quite far away. I'm just looking at the mouth, and it's looking at the teeth. It looks they're not sharp teeth. I don't know if you can zoom in on this. I might need to get some forceps just so I didn't hurt myself. But these are very round teeth, quite like a gummy shark. So let's have a look at the underside of the shark and see what the fishmonger has left in and what is still there. As I said, we've had a look at the teeth. We've had a look at these nares in here, and we've had a look at these ampullae of Lorenzini. Now, if I draw back the innards, unfortunately pretty much all of the internal organs have been removed, which is, we can see a little bit of liver there, and we can tell from the colour, it might even be a spleen, it's very difficult to tell. And then we've got this organ here. Now, it does look like a heart, and the best way to tell, to be honest, is to look at what's going in and out of it. So if I look here, you can see a very elastic pink structure, and that is definitely a vessel, okay? Specifically the aorta. I don't know if is that visible there? Yeah? Okay. There's not much else, unfortunately, apart from but you can get a very good view of the muscle, which is divided up into myotomes. And there are a lot of them. Remember, sharks, very powerful musculature. And then if we look down here, we can of course see the cloaca, which is where everything exits the shark in terms of feces and reproductive things. So, first thing I'm going to do is just snip out the heart, and let's take a closer look at it. So let me get my iris stitch scissors and forceps. Here is the heart of the shark. You can see this lovely aorta here, very thick elastic vessel that outputs blood to the rest of the body, and then this thinner one here will be going into the heart. The heart of a shark has two chambers. It has an atrium and a ventricle, and here you can just see this atrioventricular valve just there, and inside you can see lots of ridges. These are the trabeculae carni. These shape the flow of the blood and decrease turbulence. The hearts of sharks also has two smaller pseudo chambers. So what we can see here is a very nice view of what I think is the bulbous arteriosus. The other chamber of blood going into the heart, which would be about here, had it not been chopped off by the fishmonger, it would be the sinus venosus. But here we do get a very nice view of this little antechamber just before blood would leave the heart in this aorta. Now, if we want to see where the gut would have been, what we can do is we can take a probe and we can stick it down the shark's mouth and see where it comes out. And there we are. It's that is indeed where the esophagus would have come out in this shark. And then the gut would have been all around here and come out at this end. Now let's have a look and see if any gills remain. So, most sharks have 5 gill slits, but there are some sharks which have 6 or 7 gills. In fact, they're so distinct that they're actually called 6-gilled sharks or 7-gilled sharks, and they usually live very deep down in the ocean in abyssal areas. But this is indeed 1, 2, 3, 4, 5 slits of gills. Okay, and there we have a very good view of the gills of the shark, often not seen. So, we have a look here. We can see they're very fine feathery structures, not unlike the gills of bony fish. And then we can also see along here on the inner edge. So this is what sharks have for a respiratory system, and it's very effective. The next thing, and probably the last thing we can look at in a shark that's been gutted like this, is the nervous system. We're going to try and look at 2 things. First, the eyes, and then second, the brain. So let's try and get to the skull first, and if we can't do that, then we can just go straight to the eyes. So, I'm going to use these scissors and snip down here. This is going to be very tough, but I'm probably better off using the scalpel. I have a number 22 scalpel here, not typically used in human surgery, and I'm going to cut away from me. Actually, no, I'm just going to cut this way. Back to those flaps and see what we've got. That's better. So I've made my cranial flap, and I'm just going to remove this bit of bone here, and now we have an uninhibited view of the shark's brain, bathed in that cerebrospinal fluid, and once we're in and we know the depth, it's pretty easy to just remove more of this cranium. So, although I've been saying bone a lot, that's just what I'm used to, and I don't actually mean bone. That's just a slip of the tongue, really, because as I say, it's what I'm used to. Sharks are, of course, cartilaginous fish, so their skeletal structures are made of cartilage. So you can see the metal behind it, and it's quite nice and bendy, because this is cartilage. We've got a very nice view indeed of this shark's brain here. Can't really see any structure at the moment. Let me see if I can tip out some of the fluid. That's a bit better. We can now see this lovely cerebral tissue here. This pale white stuff, and that's because neurons are, of course, surrounded by myelin, which is a nice kind of fat. And I can see some structures here, but I'm not sure they'll be visible to the camera. So what I'm going to do next is look at the eyes. In taking out the eye, I should be able to see the optic nerve and be able to trace it back to the brain that we've already got a view of. Now, if you've ever seen a shark in an aquarium, which I'm sure you must have, you'll know that sharks have a lovely nictitating membrane, which is what this is on here. Oh dear, that's a picture of squirting out. Right, so I've clearly made a mess of this. I've perforated the cornea, we've got aqueous humour bubbling out, it's a right mess. So what I'm just going to try and do now is just cut my losses and get out the eye, because what we want to see is that optic nerve. So this is going to be a bit messy, I'm afraid. Oh dear, oh no, oh dear, this is, sorry. Oh, yeah. Okay. Actually, this is not a bad dissection of the eye here. Can you see that? So we've got here, this pearl is the lens. Look how beautiful and round that is. That is what a shark's eye uses to focus the light onto the back of the retina.
Wait, it's not in focus.
Oh,
perfect, stay there, go.
Okay. So we've got quite a nice dissection of the eye here. You can see the clear window at the front of the eye, that's the cornea, and the white of the eye, which is very tough as we just saw, is the sclera. The black stuff is the vitreous, which is transparent in the living shark. This is the lens. You can see it's slightly squishy. I've just squeezed off the lens capsule. You can get a very good view of the different layers of the lens. So you can see there's a transparent cortex and an inner less transparent core. So I'll just rotate that around, hopefully you can see the different layers. So apologies for the mess, but we now should have a good view of the orbits. Let's just pull this out. This long thin white thing is of course the optic nerve, which was attached to the back of the eye, which is what I'm holding now, this messy stuff. So we have the optic nerve there. The other nerve I'd like to draw your attention to runs along the floor of the orbit, and it's this thick one here, the infraorbital nerve, which has two branches, as you can see. Now this thicker one here is the maxillary nerve, and that's because it supplies the maxillary area with sensation and motor, so it contributes to the opening of the mouth. Now the other branch of the infraorbital nerve, which is this thinner one you saw here, that is the buccal nerve, and that's because it supplies the buccal cavity. The buccal cavity is the inside of the mouth, so it supplies sensation to the inside of the mouth. Now the infraorbital nerve is actually a branch of the maxillary division of the trigeminal nerve, which is one of the cranial nerves, i.e. one that goes directly into the brain. And the trigeminal nerve in sharks actually has 4 divisions, and that's because the first trigeminal studied was in humans, where it did have 3 divisions. I just want to end by reiterating that these are beautiful creatures and I'm not going to eat it, neither should you. The only reason I bought this was for dissection. And that's all I have for you today, so thank you for watching, and if you have any questions please leave them in the comments.
Oh, bollocks.
In any way condone
Stop!
Running.
No, there's a bit at the end. Wait, tell them what happened. I'm recording.
Okay, so we were struck on by a sudden hailstorm which has now abated, so let's go back in. Oh, it's raining! Why? Why? No, that's insensitive. Doesn't do that. So we've had a look at the shark and everything we could have that's still left inside it, and we've seen how we've seen the innards of this remarkable creature, which we should do our best to preserve.